rolling along in the lab, and then we stumbled a little bit with some instrumental issues. But I'll present what we have so far. Basically, what I'll talk about is what we all know, so it's kind of a foolish section, so I'll skip through it. But why it's important to look at pharmaceuticals and other endocrine disruptors in some of the major rivers in the Midwest. Then I'll talk about the scope of our project, some of the sampling, extraction, analysis, but not much because Teresa will be covering a lot of that. Some toxicity testing and some ending comments. A lot of this stuff has been published in either the late 90s or early 2000. It's estimated that between 30 and 90% of pharmaceuticals that we ingest exit our bodies unmetabolized or intact. Those then get sent through wastewater treatment plants. Some get eliminated, some remain. Or we could be looking at feedlots where we have veterinary antibiotics that are released and then make their way into surface waters. The interesting thing about the pharmaceuticals and their release is that they are released or discharged in as high amounts as pesticides are, yet they're not regulated such as pesticides are. We know that they're prevalent in waters, and actually it kind of struck me when I was listening to a talk earlier. Aquatic toxicity testing always assumes either a 48 or a 96-hour dose or exposure, which is highly unrealistic if you're looking at pesticides, because it's usually a pulsed exposure that you're exposed to. However, when you look at the pharmaceuticals at the trace levels, you are getting that realistic dose of a chronic exposure. So someone asked, why would you even think that there would be a seasonal effect of the levels you find in pharmaceuticals in river waters? Because you would expect people are not seasonally taking pharmaceuticals. However, some have found that there is a seasonal effect, and most likely due to the fact that there's less degradation in the wintertime, the hydrolysis rates and whatnot are slowed down. And then doing some literature, we found that there is an extraordinary amount of pharmaceuticals. This is from, I believe, statehealthfacts.org, and it's looking at the retail dispensation of prescriptions in the United States in 2006. We see higher levels sort of in this area. I just drew the line. So these are the lowest amounts that they're sold. And then these are the top 15 states with sales. Illinois ranking about 6th, 7th, 8th, somewhere in there. And then you see Missouri and Tennessee tailing. But this is the one that caught my eyes. If we look at the per capita distribution of pharmaceuticals, the Midwest is receiving a mother load of those pharmaceuticals. So we got involved because we were working with persistent organic pollutants, and then endocrine disruptors, and then pharmaceuticals got added, and people just kept adding, and we just kept saying, sure, sure, sure. But the pharmaceuticals is where we're at right now, and so we wanted to look at different levels in either tributaries or just alongside the river, Illinois River, or different sites in the Mississippi River, either above or below the confluence of the two. And then take those results and look at individual pharmaceuticals and then mixtures. Now, obviously, the mixtures are unyieldy with some 60 to 80 compounds showing up in different samples, so that would be Pandora's box and not realistic. At first, we were going to look along just some of the feedlots that we've sampled before. We used to look at buffer strips and how they ameliorate runoff, or in some cases they have acted as the actual source. We wanted to look at weekly sampling first, then maybe biweekly if we're seeing some constancy. We intend, remember this is preliminary studies, to do some hourly sampling for 48 hours. Initially taking individual samples, then taking some composites. hoping to get the most realistic exposure concentrations and scenarios, however, realizing that there's no way we can recreate the multitude of combinations. Again, I think I already said all this. Above and below the conversion in Grafton, looking at some tributaries, some rivers that are running into lakes that have already shown decreased ecological activity, and then some pre and post water treatment facilities. There's a number of scenarios playing out because there's a number of different students that are interested in looking at this, and so I just keep adding to the possible scenario that they can look. The main thing that we're working on is developing the extraction technique. Right now we've basically we've got the parameters down for about a third of the compounds we're looking at. These are the pharmaceuticals and some of the EDCs. There are multitudes more depending upon which screen and what extraction method we use. The one that's predominant or prevalent in the, or the couple in the literature has to do with the hydrophilic lipid balance, the HLB Oasis with the same resin that was in the passive sampler we saw earlier. That's where we're going to start using 1 liter samples. Depending upon the method, they vary the pH between 2, 7, and 9. And of course, that's going to dictate which compounds we expect to see because we'll basically hydrolyze some, not hydrolyze others, cause some to elute. But at least we're going to start, we'll start neutral and see which way we go. A lot of these we're going to have to take paired or triplicate samples just so that we can analyze them differently. Briefly, the analysis is with the Varian LC-MS-MS triple quad, where we're looking at electrospray ionization, not with the persistent organic pollutants, but with the pharmaceuticals and some of the endocrine disruptors. We've basically got down to 50 on all of those. That's part per trillion. 50 part per trillion on all the ones I showed. There's another variety, or about 20 more that we're down to 10 part per trillion. And as we keep pushing our detection limits, we keep pushing Varian, and so they seem to like to hang out in our lab. Just so I had results, because I will have no other results, some of what we've found is just the operating parameters for the MS-MS, where we're selectively isolating one ion for the molecular ion and then breaking it down and then analyzing and quantitating based on the daughter ion. I won't go into this. I'll expect traces. So again, we haven't done this yet. We have with insecticides, looking at individual compounds, looking at a few combinations, and even when we got into 3 different chemicals, the combinations and concentrations started to get out of control. And so the reality of being able to do that with the pharmaceuticals is unlikely, but we're going to do our best to see what we can find. We will look at seasonality just to see if that has an effect or not in the toxicity testing using different levels. That would be the seasonality effect there. What are we going to look at? Well, first of all, we'll do some static renewal 96-hour tests. We're going to use Daphnia magna and some, I guess I didn't put it up there, some pulsed exposures, but we don't expect that to be very relevant. We just wanted to look and to compare that to some of the the pesticide data we've generated. But what we'll be looking at essentially is mortality as the main endpoint, and then for a period of 21 to 28 days after that, looking at reproduction, looking at molting, etc. Long-term indicators of ecological change. And just on an end note, I guess I started by saying this was pre-preliminary, so we're very early in the process, even though we've been working with these for about, I'd say, a year. We are gearing up our water extraction methods. Actually, we started this week and are moving forward on that. And another, I guess, ecotoxicological question has to do with the bioavailability of these contaminants. If they're present in the water, does that mean there's an effect? Excuse me. We've got some some 178 turtle blood samples that have been collected on the Tennessee River over a 5-year period by Tom Wilson down at UT Chattanooga that will be screening just multitudes of ways with the persistent organic pollutants, endocrine disruptors, and the pharmaceuticals. We tried to optimize on 2 of the 3 initially, and now we realize we're going to have to optimize on the endocrine disruptors and some of the persistent organic pollutants, and then just know what our extraction recoveries are of some of the pharmaceuticals, given that that wasn't what drove that study. And then continue looking at some biomonitoring or going further up the food chain into some fish species. And with that, I'll conclude.